Good morning and good afternoon.
Hello,
Hello.
Beno. Do you,
Hello.
do you hear me?
Can you hear me? Super
Yeah.
Anna.
Hey,
Very happy to see you.
nice to meet you.
Nice to meet
Hello,
you too.
everyone. I'm happy to start the second block of our webinars. The key topic is biomanufacturing, bioprinting, and happy to start it with Beno Juarez, who is a pioneer in digital fabrication technologies in Latin America, who is co-founder of the first FabLab in Peru, Lima. And yeah, Beno, please start your lecture.
Thank you so much, Super Anna, and thank you, my friends, for this opportunity to share these ideas and challenge. Well, I come from this area, from Satipo in the Amazon rainforest in Peru. This is part of the biggest rainforest in the world, and this is a very exciting place where you can be stimulated by nature, by shapes, by colors, by rivers, etc. It is a really beautiful place, and also it have an extreme conditions. You know, well, part of the challenge in the jungle is the deforestation. We are losing the jungle, we are losing the woodland. Also, even if this is a very rich area in terms of biodiversity, it's also one of the poorest areas in terms of social development. So this is part of the challenge. This is the context where I grew up, and also the context that inspired me to explore this new kind of technology for a sustainable world. So we have many challenges in the Amazon rainforest, but also many opportunities to learn from nature, to learn for future technologies, sustainable technologies, etc. So part of my life is FabLab network, thanks to Vicente Guayar. No? In 2009, I been joined to this amazing network. And also, um, I am part of the FabLab network, the Latin American network. And a few years ago, also be part of this amazing program. It's How to Grow Things, no? Led by George professor. It's the future of digital fabrication technologies, how to control the shape or the material productions, and many different things that we are going to explore in the coming minutes, integrating biotechnology with digital fabrication. And also I was honored to be part of the Chukov lab in 2019, 18, sorry, with different exploration in the lab, how to 3D print, and also how to connect that with cities and how to impact in the city, etc. So this is part of my life or of my background. But I have some questions, some main questions. I think everybody have main questions that are thinking every day or constantly, and try to get these questions. So in my life today, it's how to integrate this biodiversity with digital fabrication tools in order to generate a sustainable impact for the world. So one of my questions is nature doesn't 3D print. So I am excited and fascinated with 3D printers and digital technology. But when you explore nature, it's a different process. No? It's grow inside from outside. And it have another kind of properties that are interesting and challenging our notion of technology, of digital technologies. And I think it will be an amazing input for different projects in this field. So I would love to share with you three challenges. And if you are interested or you are maybe developing or you are involved in these fields, I would love to partner with you in order to explore more deeply this challenge. Well, in traditional digital fabrication manufacturing, we have as a challenge, for example, how to reprogram a structure. As you can see here, this is part of the challenges of today technologies in digital fabrication. How could we integrate different materials inside, for example, a 3D printer or one single system for manufacturing something? How could you work with different materials? Now, this is part of the other challenge. And the other big challenge is how could you recycle material? You are printing, we are using a lot of material, but how could we integrate this process inside the same system, inside the same machines? So in biotechnology, we have amazing opportunities. for example, one of the challenge or one of the opportunities that we have, it's you can use biologies in order to control the growing of an structure. Also, you can use biology in order to programming material and generate many different materials as you want. With one single input, you can generate many different materials. And also, you can bioregenerate your structure. You can generate, you can print or make something that will self-generate if it's needed. So this is part of the opportunities and also challenge. Let me share you some technology that could help us to address these opportunities, these challenges. In the first field, in the first area, is how to control the growing of one structure, no? As we can find in nature. So this friend is Pambil, is the walking tree. It's very common in the Amazon. And imagine a tree that can walk, that can move in, I don't know, 10 years, maybe 5 or 6 meters. No? Yeah, that's interesting because I don't know if you saw some tree walking in the urban area, but what's interesting in this plant, no, it's the roots grow outside of the ground. So when the roots grow, and in the Amazon, the soil is very unstable because the level of the water every year, no, the water rises more than 2 meters, so it generates like a fluctuation. Okay, so this fluctuation generates a very unstable growth. So in some moment, the plants are falling down. No? So when the plants feel that are falling down, it will grow more roots in this direction. So at the end, the plants looks like are movement, but at the end are growing. No? It's a very interesting system. And if you explore what is happening inside the cells of these plants, it has small sensors. No, it's a small sensor. It's imagine that you have a balloon, and inside the balloon you have a small ball. So doesn't matter where you move the balloon, the small ball will keep in the bottom of the structure, in the bottom of this balloon. So these plants have this similar sensor, so it doesn't matter where it moves, it will recognize where is the direction of the gravity. So imagine to use this technology of movement to apply, for example, in another fields. No? One of the biggest problems in the Amazon is that this kind of houses have to regenerate the pilots or replace the pilots every 2 years. No? It's a huge effort for these people. Imagine that you can design a structure that can self-regulate this position according to the water level of the rivers and following this logic of the pumping. So this is on a big scale, but when we go deepless, we have different properties that we can use in order to control the movement of microorganisms. So we could use different stimulus like light, chemical stimulus, or electrical, or gravity, for example. And also we can generate different reactions. No? We can control different reactions, for example, movement or growth. No? So if we mix, for example, light with movement, we will have phototaxis, no? Or maybe chemical tropics, etc. So this is important because with these stimulus and reactions, you could control a shape of a biostructure. So this is what we started to explore. In this case, we made these small labs in the FabLab and starting to control the growing of fungi. No? First, we explore with some bacteria, no? E. coli, and putting, in this case, electrical stimulus in order to control the direction and the position of the bacteria. And here with fungi. What's interesting, you can find here two fungi grows with the same time. So at the top, you have fungi with electrical stimulus, and in the bottom, just release fungi and growing happy. So what's interesting here, with in the fungi that grow without any stimulation, they grow easily and horizontally. But with this kind of stimulus in the bottom, and the top, sorry, the fungi started to grow in vertical. So with this kind of stimulus, imagine you can mixture this horizontal and vertical growth in order to generate another more complex structures. No? There are different explorations in these fields, how to control the movement of the growing of microorganisms using digital technologies. No? For example, this is one experiment that we generate, inspired in the professor Nina Tanton. You can search in Vimeo, no, Paramecium HTGAA 2015 to look for this interesting experiment where you can use your joystick and you can control the movement of the Paramecium. We did that during this program. Well, this is part of the properties of this microorganism. So this is the starting point. You can use this technology, you can use these properties in order to generate another kind of structures. No? So our goal with this field, no, research field, is how could we programming a 3D structure and how could we reconstruct that layer by layer, partner with these microorganisms or different microorganisms. In order to have more, have a deepless control, you can also hack, for example, in this case, the bacteria that we use is an E. coli, and E. coli have different genes, and you can introduce inside this the field of the genes of the E. coli, for example, the property to follow UV light or infrared light. So with these properties that you can put inside the bacteria, and you can use in this case for this goal, you can introduce synthetic biology inside your research, no? And you can control the growing that in one direction, the growing in one direction, and also the dispersion of the root of the structure. No? With these two properties, you can start to explore how could you create different shapes. No? So this is part of the exploration with synthetic biology and future 3D printing. And it means to hack the bacteria, no? To insert inside this bacteria this property to follow light or to change direction. So the second challenge, this is part of our also exploration during in the Flooring FabLab that we'll talk at the end. No? It's how to programming material. You know, well, when we use a traditional printer, for example, yeah, we can use different materials but in similar properties. But what will happen if you can program in the material that you want and using the same inputs? No? And also, what will happen if you can send not only material, you can send information through the material? This is part of my biggest challenge. No? How could we recreate an artificial gland? No? What is that? Imagine that you have a flow, no? If you send a flow, what is interesting? Let me come back here to the gland. What is interesting in gland? This is part of our system. No? In our system, we use different hormones in order to control different reactions. So what is interesting in a gland? You are not only sending information; you are also sending material. You send both, no? You send information and material. What happens if you can use this logic to create a structure that could make itself, no? Not by the external forces, but by internal forces. No? This is part of our challenge. So we started to explore how to control a fluid, and then how to send information inside this fluid and create a branch with that. This is part of our challenge. We didn't get yet, but I would love to share you that. No? For example, when we started to explore with some materials that we find in the jungle, this is a very common material, it's a bee wax. No? With these bee wax, some fluidics, no? We started to create a pipe. No? So after to create a pipe, injecting a liquid inside this mixture of different biomaterials, Then we started to send different properties inside this liquid in order to flip the liquid, in order to generate a branch. No? So this is part of the exploration using different materials and different temperatures and different pH, etc., in order to generate this change of the linearity of the structure. No? So it's a small pipe, no? And with this information, the structure starts to change. So we create this machine in order to control and automatize this process. And then we started to explore with biotech, how could we reprogram that material that the machine are printing? So let me explain you a little bit this graphic. We have here 3 different growth. Here we have bacteria, no, that will detect the level of salinity of the liquid and outside liquid. No? And then, according to that, it will produce different proteins, and these proteins will control the production or the replication of another bacteria, no, according to these proteins that these previous bacteria have produced. No? So what happened here is you have a control system where you introduce a level of salinity, and this level of salinity could stimulate many different inductors, no? And these inductors will follow, no, continues stimulating the other growth. For example, in order to produce keratin, in this case is keratin, it's a very hard and strong protein, no, that you can find in mollusks, for example, or a collagen, it's more flexible, or a fibrin, no, it's a mixture. It's flexible, but it's also strong. No? So with this system, our target, our goal, it's using one single input, you can switch inside and bioproduce different proteins and materials and generate a smooth and continuous material, but with different properties. No? So we started to explore with chemical reactions and also with bioreactions. This is a key part to address this goal. It's how could you design a pathway that connect one input with another output. The input what you want with the output that you want. This is not happening necessarily in nature, but you can create artificially connections in order to, for example, in this case, how to convert cellulose in chatan, no? Or chitin. The cellulose have a pathway, no, and you can map this pathway. But the other hand, you have chatan with another pathway. So, but you can, for example, find as a common fructose, no, and fructose, you can create this integration, so you can recreate that in artificial system or in a bacteria, for example. How could you introducing cellulose will produce chatan? If you want to explore more details, I suggest this web page. You can go here, no, an open source Cobra. It's a community development code based for constraint-based reconstruction and analysis, and you will find a lot of communities around this this topic. So we are using this kind of technologies in order to produce also our own food. No? This is part of one exploration I started a few years ago, where we start to explore what will happen if we can have our human microbiome to produce the food that we need every day. Imagine that you have in your skin a lot of microorganisms that you can hack also, and you can use this for your own nutrition and also for your own protection of the, I don't know, say UV, etc. So this is Nutrigene, no? It's an exploration about how to switch from the macro agro to the micro agro in this kind of machines that can, this is a small bioreactor where you can put microorganisms and can produce any food that you need, in this case, any nutrient that you need per day. No? So it is an integration of digital fabrication and also biotech with synthetic biologies. We use microalgae that has been synthetic modified in order to produce high concentrations of different vitamins. And the regulation of this comes from the input, no? In this case, different inputs that you can send to the algae in order to switch between one vitamin to the other. So this is part of the development and part of the prototypes that we was creating, and yeah, different models that we did in order to produce this in your house. No? So imagine that you wake up today and then you have your phone, and your phone will control, according to your stress level or your different nutritional levels, the production of your vitamins. And well, at the end we have this search challenge, is the regeneration. Regeneration is extremely important for nature. What will happen if we can introduce also these properties in our products, no? in our 3D printed objects or whatever we are developing or designing. So in nature, in the Amazon, we have this amazing plant. It's ayahuasca. I don't know if you have listened about this. It's like an hallucinogenic, but also it's a sacred plant. No? Shamans, people, or indigenous people use to visualize the future. No? It's a very respectful plant in the Amazon, but also have interesting properties. If you go deeply to the ayahuasca, you can see that it have as different plants, no? The xylogen and phloem system. These systems are collaborating between them, because one of the systems bring them the structure, no? It's a strong system, but the other bring to the plant the flexibility. And with the external tissue of the plant, it's carried, the internal tissue will repair. So this is a very interesting symbiosis in between this kind of tissues that the plant have. It is a very regenerative system. All the plants have this, but in the case of ayahuasca, it's interesting because it's super fast, this regenerative system in between these two tissues. No? So one of amazing technologies that could help us to address this goal of regeneration, it's synthetic minimal cells developed by Kate Adamala. She's an amazing professor that have this webpage. You can also look for that, no? Protobiology.org, and you will find what synthetic minimal cells means, what are all the potentials, and also she have this community. You can make, you don't need to use a complete cell in order to prototype all these kind of systems. No? What is interesting in the Kate Adamala theory, she said, if you use a complete cell, you are spending energy in functions that maybe you don't need for this goal. No? You can just take the parts of the cell that you need and encapsulate in an artificial cell and generate what you want. No? If you want to produce something, yeah, just use only the components that you need for this goal, but you don't need to use everything. No? So all this exploration you will find here, and you can, of course, contact Kate Adamala if you want to go for more details. So we use, in a theoretical exploration, these principles in order to generate a self-regenerative structure based in the silkworm. No? The silk, it's an amazing also material, no? And as you can see here, similar to the ayahuasca, it has two layers. No? One very strong layer, it's a fibrin, and one smooth layer, in this case, is the sericin that will protect the silk. No? So in this synthetic minimal cells exploration, we start with genetic level. No? So what kind of genes we need to control the production of one or other protein, no? Sericin or fibrin. So, and this is our goal. No? The function of the sericin is protect to the fibrin from external influence, as for example the light, because light will degrade the fibrin. So if you are in a context where you have a lot of light, you will want to produce more sericin. But if you have not too much light, you don't need to spend too much resources in sericin. You will prefer produce more fibroin because it's one of the main properties of the silk. So according to this path and this goal, we design inside the cell this is an artificial cell that will need protection. No, it is a lipid membrane that will protect the cell and will encapsulate all the functionalities that the synthetic minimal cell needs. And according to the level of radiation, no, you will produce more or less sericin or fibroin. This is the principle, and part of the technology that could help us to address that is synthetic minimal cells. Okay, and finally, I would love to share with you the platform where we are exploring and where we are connecting all these ideas. This is a Flooring FabLab Amazon. This is a project that, well, I am personally involved for 5 years or more. And the address is how could we incubate future manufacturing inside the Amazon, inside the green hair of the world. We have a lot of searches and friends that are involved also in this project. One of the goals is how could we create a regenerative structures. Imagine that you have, this is part of our research also, exploring what kind of microalgae do we have in the rivers, how could we use these microalgae to generate biopolymers, no? And how could we use this biopolymer in order to generate, for example, the facade or some structures of the flooring lab. And of course, after that, it will come back to the river and bioregenerate, be part, of course, of the landscape, and we could create this self-regenerative cycle with nature and with the environment to make this go. Well, it was part of our challenge, of my challenge, and I would love also to share with you ideas, and if you are interested in go deeper in one of these ones, or maybe in all of these challenge and opportunities, well, I will be happy to share with you more ideas. Thank you so much, friends.
Thank you, Beno, for the great lecture. Maybe we have some questions. Okay, while we are waiting the question, I can ask also, maybe you can have another examples of connection between material. No, for example, I wanted to ask how we can use these cells in facades, for example, in building architecture in big buildings, how we can translate it in large scale. Maybe you have more examples.
Yeah, more example in how to change the scale, you say? Sorry, I could. So, could you repeat this part, please? More.
Sorry, something with his voice.
Yeah, so you say more examples in how to scale.
How to scale these cells into architecture, to buildings, maybe, yeah.
Oh, yes. Yeah, this is a very important challenge also. No? The scale is extremely important. No? We are talking in a very small scale, but we can find many different examples. One of my favorite examples, for example, in regeneration is Uros Island. I don't know if you have listened about that. It's in Titicaca's Lake in between Bolivia and Peru. Mm-hmm. All the island, it's made by a plant that grows in the lake. So people take this plant, make the island, make boats, make the house, make the furniture, make everything with this single material. And what's interesting, after 1-2 years, it comes back to the lake and then return. No? So this notion of cycling, it's extremely important. Today, Professor Juarez, for example, he's a master in genetics, are exploring similar ideas, no? But with microorganisms, with bacteria that maybe take or fix the CO2, no, in more strong concrete or another kind of material. So I think materials exploration is the field that could join, no, that could integrate the small scale with the big scale. No? Different kind of material, for example, with bioluminescence, or with that also can change colors, change temperature, because have inside, I don't know, microorganisms that could help us with these fields. No? So materials could be one common point in between the micro scale and the big scale, fascinating field of exploration. And the other is the principles. No? I think when you explore the principles, you can also scale these principles with another technology and with another system. For example, maybe I know one simple example about that is, okay, if I am looking for a regeneration system, where I have to change my notion that I am designing a fixed structure. I have to change my notion that the architecture is a liquid, no? It's a fluidic. So I am designing a big fluidic system with different characteristics, and how could I integrate one fluidic with the other, and how can I control constantly sending information? Imagine that you have a big structure where you can send one kind of liquid, I don't know what, no? Maybe water, maybe, I don't know, a mixture, maybe oil. And if you have a liquid, it will be easier to control the different properties, no? So this is what happened in nature, no? You have a structure, but it's also a liquid, no? Constantly. So if you have this kind of system, I think one important learning lesson from nature, it's material is not only material, it's also information. No? How could you mixture that? But it in itself. No? So if I have this pen, I have not, this is a material, but it's not, I cannot send information here. No? And if I have my phone, no? It's the opposite. No? It's a lot of information, but the material also will not change. No? So how could I mixture in order to, for example, use that and send some material, some information here to change this linearity for a sphere. No? If you want to explore something like that, we have to switch from fixed material to liquid or liquid materials. Yeah.
Okay, so great. It's a really big challenge for architects, for engineers who explore nature by materials. I think it's very interesting for us. But unfortunately, we have more questions, as I can see. So I think we can finish our meeting today. Thank you so much, Beno, again. See you in the next lecture the day after tomorrow. Yeah, we'll be also connected with biomaterials, biomanufacturing, with bioprinting, and also we will have one lecture more next week. And after this, we are planning to have like practical school. And
Whoa,
I hope technology will join us. So see you. Thank you so much.
Thank you so much. See you soon.
Bye,
I would love to share with you. Yeah, just to finish, I think this is these are three challenges, but I think the biggest one is this one. It's the self-replication. How could you create something that will self-replicate? This is the biggest challenge that we are exploring. Please, any questions and comments and partnerships, that I would love to share with you more of this and to join your adventures also. Thank you so much, Super Anna.
Thank you. Bye bye.
See you, friends.
Bye bye.